Alright everyone, welcome back. In this lecture, we are starting IMAT Biology Lecture 2, which covers cell structure, cell membrane, and transport mechanism. I have the PDF open on screen. I will teach directly from this PDF. If you follow only what I highlight, circle, and draw, this lecture alone is sufficient for IMAT cell biology. Before touching the content, I want you to understand how IMAT asks questions from this chapter. IMAT almost never will ask definitions. Instead, it will test three things. It will test comparison between structures. It will also ask what will happen if a structure is missing. And also, there will be some elimination type questions using correct biological fact. Keep this mindset while we go forward. Here comes the first topic, cell theory. Three fundamental core statements. The first one is all living organisms are composed of cells. Then cell is the basic structural and functional unit of life. And the third statement is new cells arise from pre-existing cells. The statements might look simple, but based on these statements, we can tell virus is not a cell, or virus is acellular, right? On the right side, we have a diagram of cell. Before diving deep into the lecture, we should point some structures that will be highly tested on our exams. First of all, you see that the cell has a membrane outside. This is called the cell membrane. And inside the cell membrane, we have cytoplasm. And in cytoplasm, you have mitochondria floating, then endoplasmic reticulum, and the nucleus is also one of the most important parts of cell, because inside nucleus you will have DNA. So the left one that we have discussed, this is a eukaryotic cell. That's why the structure is a bit sophisticated. And on the right side, we see that this is an example of prokaryotic cell. In prokaryotic cell, the DNA is naked and it is circular. If we study the prokaryotic cells, we will find all these characteristics. First, the example will be bacteria, and the prokaryotic cell will not have a membrane-bound nucleus. We have seen in the diagram that the genetic material or DNA is not enclosed by a nuclear envelope. It is circular, that we have also covered, and naked, deoxyribonucleic acid. It doesn't have any histone protein, and it doesn't have any membrane-bound organelles. All the organelles are absent except ribosome. But the ribosome is 70s compared to the 80s ribosome of eukaryotic cell. You see that ribosomes are responsible for protein synthesis because an organism cannot survive without the help of protein. And what about the cell wall? It does have cell wall. This is very important to consider. And the cell wall is made up of peptidoglycan. And this peptidoglycan provides the shape and protection against outside environment. Some people might think that prokaryote or prokaryotes don't have mitochondria, so it shouldn't do any respiration. But that statement or that assumption is incorrect. Respiration must be present, and respiration occurs on the cell membrane. If you draw a bacteria, and let's say this is the membrane, and in this membrane you will see the respiration taking place and ATP production too. Okay, and another important thing is that the smaller size is an efficient diffusion medium without compartmentalization. If we think about the eukaryotic cell, you see that the eukaryotic cell is advanced, complex, or sophisticated. As a result, eukaryotic cell has compartment for appropriate or proper functionality, but the prokaryotic cell doesn't have any compartment. As there is no compartment, the diffusion becomes efficient. What about the cell division? Cell division will be by binary fission. That means that, let's say this is a prokaryotic cell, there will be a furrow in between the cell, and finally there will be 2 more daughter cells, and this process is called binary fission. What are the IMAT traps that you shouldn't forget? We have already mentioned that prokaryotes must have ribosomes, but the ribosomes would be 70S. Prokaryotes do carry out respiration despite lacking mitochondria. Where does the respiration take place? Cell membrane. And organelle doesn't mean that an absence of organelle doesn't mean absence of metabolism. This is very important. Why these 3 statements are important? Because they will be tested on true-false type questions. Now moving on and going to eukaryotic cells. Eukaryotic cells can be animal cells, plant cells, fungi, or protists. What are the structural characters? The opposite characters of prokaryotic cell will be seen in eukaryotic cells. For easily memorizing it, you will remember all the positive and higher advanced characters are present in eukaryotic cell. That means that the nucleus will be true nucleus because the nucleus will have nuclear envelope, nuclear pore will be present as well, and also chromatin will be present and nucleolus will be also there. When we are talking about the prokaryotic cell had circular DNA. Compared to that, the eukaryotic cell will have linear chromosome, right? And the DNA would be like this in a linear fashion. And all the membrane-bound organelles are present, including mitochondria for respiration, endoplasmic reticulum for protein and lipid synthesis, Golgi apparatus for packaging, and lysosome for phagocytosis or enzyme-induced autophagy. Then ribosomes must be present, but this ribosome would be 80S compared to the previous prokaryotic cell. The cell size has become larger, and that's why it will require compartmentalization to maintain the efficiency of diffusion. All right. So I think we are easily able to answer questions if we remember that prokaryotic cells doesn't have anything except a nucleate region and 70S ribosome. On the other hand, eukaryotic cells have everything that you can imagine. And if we talk about the cell division, prokaryotes did have amitosis or binary fission. On the other hand, this eukaryotic cell will have mitosis and meiosis, right? And as it has mitochondria, respiration will be here, and ribosome will have protein synthesis function, and it will have compartment because compartmentalization will increase the efficiency by separating cellular processes. And so the prokaryotic and eukaryotic comparison will be very easy for us to understand right now. For prokaryotic cell, we will put minus, and for eukaryotic cell, we will put plus, right? We see that everything is absent. Deoxyribonucleic acid is circular and naked because it is not inside a nucleus. On the other hand, the eukaryotic cells DNA will be linear and it will have histone protein. What do we mean by histone protein? When we draw the DNA, this DNA will have some bead-like structures. Those are histone proteins. They help in organization of DNA. Because DNA is very long and we need to organize it properly, the organelles, all the organelles will be absent, and in case of eukaryotic cell, organelles will be present. And ribosome will be 70S, and if you break it down, then the 70S ribosome will be, in case of ribosome, we already said 70S, and if we break it down, it will be 30S plus 50S, and this S represents Wedberg unit. In case of eukaryotic cell, the ribosome will be 80S, and if we break it down, it will be 40S plus 60S. Don't get confused. These two units are actually subunits. 50S is large subunit, 30S is small. And in case of eukaryotic cell, this 60S is large and 40S is small subunit. All right, moving on. Respiration site, we see that prokaryotic cells don't have mitochondria. That's why the respiration will take place in cell membrane. On the other hand, the respiration will be inside mitochondria in case of eukaryotic cell, and the process will be sophisticated. Cell wall in case of prokaryotes, cell wall must be present and it will be made up of peptidoglycan. In case of eukaryotic cells, if we consider the plant cells, plants will have cellulose and chitin will be found in fungi cell wall. And remember, all the animal cells will lack the cell walls. What about cell size? Obviously it will be smaller and this will be larger. The cell division we have covered. This will be binary fission in case of prokaryotic cell, and in case of eukaryotic cell it will be mitosis. Now moving on to our next section, which will cover all the cell organelles. You don't need to remember the definitions, you just need to remember the functions properly. If you remember the functions, let's say one organelle is absent, and if one organelle is absent, what can happen? You can easily find out, right? So that's why it says that it will test functional failure, location of processes, and true-false statements. First of all, let's study nucleus. Before studying, let's look at the structure of a nucleus. You see that nucleus is somehow circular, and nucleus has nuclear envelope surrounding the other structures, and inside the nucleus you will see that it has nucleoplasm or jelly-like substance which acts as a matrix, and in the nucleoplasm you will see another circular structure that is called nucleolus, and this nucleolus is responsible for making nucleotides. And in case of the cells which are rapidly dividing, the nucleolus will be large in size. If we look closely in the nuclear envelope, you see that there will be two membranes. One will be outer membrane, another one will be inner membrane. And in between the structures, you see that there are pores present. Let's say this is a pore and this is another pore. What is the reason behind these pores? Inside the nucleus, you will see some nuclear material or nuclear reticulum. Nuclear reticulum, we can also synonymously say chromatin. And if we zoom inside chromatin, you see chromosomes, and chromosome means DNA histone condensed. All right, so inside nucleus you will see DNA replication. That means that inside nucleus DNA will make DNA, and from DNA RNA will be made, but this RNA will come out of the nucleus through this nuclear pore, and coming out of this nuclear pore, it will be ready to participate in translation, and ribosome will help in that process. So let's pack up everything that we have learned and let's mark up the sentences. We have said that nucleus has double membrane, and that structure, double membrane structure, is nuclear envelope. It has nuclear pores, and nuclear lamina maintains the nucleus shape, and chromatins are made up of DNA and histone proteins. We have covered all of them. What are the functions? We have said inside nucleus you will have DNA, that is genetic material. That's why it says that it will store genetic material, and nucleus will control all the cell activities by regulating gene expression. Why do we say this? Because when we draw DNA, we say that there are certain parts of DNA that gives the order for making protein, and that functional unit is called gene, and gene gives codes for protein synthesis, right? And the third one, it rightly says that the nucleus will coordinate DNA replication before cell division. Okay, and the nuclear pore, I have already told you about the function of nuclear pore, and let's see how the language will be in the question. It will allow selective transport between nucleus and cytoplasm, true, and it will also permit the exit of messenger ribonucleic acid, that means mRNA. There are various types of RNA, and among them mRNA is an important class, and mRNA works as a template for protein synthesis later on in the translation process. This mRNA will sit in ribosomal subunits, and mRNA ribosome together with tRNA will make protein. Okay, and the final one, it will permit entry of proteins required for transcription and translation. We have covered this and we have said this again and again. What are the IMAT traps that you need to take into consideration? The first one is that protein synthesis does not occur in nucleus, because we have said the replication, the transcription occurs inside. After the transcription process, RNA is made and RNA exits through the nuclear pore and it comes to cytoplasm to make protein or to perform the translation process. That's why it is said that protein synthesis doesn't occur in the nucleus. The second statement says that messenger ribonucleic acid exits the nucleus through nuclear pores. This is a trap and this can be asked in true-false statements. And we have said that the cells who have high division rate or protein synthesis rate, they will have prominent nucleolus. We have covered this too. And what about mature red blood cells? Mature red blood cells will not have a nucleus because it needs to maximize the hemoglobin space. When RBC is in the primary stage, this is called erythroblast, and in that erythroblast nucleuses are present, but as it matures the nucleus vanishes because it needs to accommodate hemoglobin, and hemoglobin will in turn carry the oxygens. Okay, now let's come to mitochondria. Mitochondria is extremely high yield. Whenever you will listen about mitochondria, you need to remember mitochondria is synonymous with energy or ATP production. Before learning the description, we need to check the diagram. You see that mitochondria has two membranes just like nucleus. One is outer membrane, another one is inner membrane. The inner membrane has some folding and this folding is called cristae. Okay, inside the inner membrane you will see matrix. All right, in matrix you will see that it has DNA. That means that mitochondria has its own DNA just like nucleus. Another organelle that has its own DNA is plastid. You need to remember that. Inside you will find granules and you will also find enzymes that will make ATP. The enzymes that are found inside the matrix are called ATP synthesis. Okay, you also see that ribosomes are present in mitochondria. That means that mitochondria can make proteins of its own because mitochondria needs so many enzymes to function properly. That's why it has its own ribosome. Okay, and just like nucleus, it also has some pores and these are called porins. Now let's match the description with the diagram. First of all, the core function, it says that mitochondria is the site of aerobic respiration. What do we mean by aerobic? We want to say that this respiration will require oxygen, right? There's another type of respiration that is anaerobic. It doesn't need any oxygen in that process, right? And mitochondria is the major producer of adenosine triphosphate or ATP. ATP is also known as energy currency of cell. The structure is written here. I'm just gonna mark the important words. It is outer membrane that is permeable to small molecules, and inner membrane is folded into cristae. Intermembrane space is for proton accumulation. This statement we will understand with the following diagram that I have for you guys. And we have also talked about matrix. Matrix contains enzymes, DNAs, and also 70S ribosomes. Very important to know. Okay. Why is there cristae present in mitochondria? Because increased cristae will increase the surface area for electron transport chain proteins. We will understand this concept too. And the second logic is that electron transport chain, if increased, will also increase the ATP synthesis, which is very important for our metabolism. Okay, so this is the electron transport chain that we were talking about. So this is mitochondria, and we have taken a slice of the membrane of mitochondria and we have zoomed inside it. If we see closely, we will see that in this membrane there is an intermembrane space, and this is inner membrane and this is outer membrane. Okay, let's take the laser pointer to explain. So this outer membrane and this inner membrane has intermembrane space. In the inner membrane, you see that we have some protein complex or enzyme complex, and there are 1, 2, 3, 4. 1, 2, 3, and 4. These 4 enzymes together are called enzyme complex, right? What they do, they actually take protons and they pump protons inside, and that protons are taken up by another enzyme. That enzyme is called ATP synthase, and with the flow of the proton, ATP synthase makes ATP. Okay, so based on this diagram, we can justify these 2 sentences. One is that intermembrane space is for proton accumulation, very important. And we have also said that if the surface area is increased, then there will be more electron transport chains, and that will be helpful in making more energy. Okay. So here's an important information that energy demanding cells contain many mitochondria. Can you name one energy demanding cell? That will be heart, because heart needs to pump a lot. Then liver is also energy demanding organ. That's why the heart cells or liver cells contain so many mitochondria. There's an evolutionary significance behind mitochondria. The presence of deoxyribonucleic acid and ribosomes support the theory of endosymbiosis. That means that mitochondria was trapped inside a cell and it then evolved into a higher type of cell, and that is endosymbiosis. Okay, what are the IMAT traps? These are the IMAT traps that prokaryotic cells doesn't have mitochondria. And mitochondria are double membrane organelles. Why it is said so many times? Because cell organelles can have 3 types of membrane. One can be double membrane, another can be triple membrane, or can be single membrane as well, or can be single membrane as well. And there are some organelles that doesn't have any membrane whatsoever. One famous example is ribosome. Ribosome doesn't have any membrane at all. Okay. And the third trap is that mitochondria depend partly on nuclear genes despite having their own deoxyribonucleic acid. This is very important. We have seen that mitochondria has circular DNA. Even after that, mitochondria will depend on the DNA of nucleus to fully function. Okay. Now, ribosome. When we were looking at the diagram of the cell, we saw that ribosomes were freely floating, or some ribosomes were also attached to endoplasmic reticulum that we will discuss later. So ribosomes, if we talk about the structure, ribosomes are non-membrane bound particles, and it is composed of rRNA and protein. Very important high yield information. Okay. So there are some important RNA types that I should mention. One is tRNA, and it does the transfer of amino acid. Another one is mRNA. Then there is gRNA that works as genetic material. Then there's also rRNA, that is ribosomal RNA. Then there is small nuclear RNA as well that helps in enzymatic function in the central dogma that we will study later. Okay, After covering the structure, let's talk about the function. We have covered and we have said that ribosomes are the site of protein synthesis through translation of messenger ribonucleic acid. This mRNA works as a template, and if we draw roughly, then we see that, let's say this is ribosome, and in ribosome we see that mRNA sits, and mRNA has sugar bases. 3 sugar bases together are called codon, and following this codon, tRNA will bring some amino acids, and let's say amino acid is brought here by the help of tRNA, and when the tRNA sits with anticodon, it will make amino acids, and the amino acids will be bound together, which will form peptide and eventually protein, right? And ribosome, you see that ribosome has 2 subunits. One is small subunit, another one is large subunit, right? So in case of eukaryota, we have said that the ribosome is 80S and the subunits will be 40S and 60S. In case of prokaryotes, the ribosome will be 70S and the large subunit will be 50S and the small subunit will be 30S. Okay. Now comparing with the diagram, let's find the statements and the logics. There are 2 types of ribosomes. That's why it is said that one is free ribosome, and this free ribosome is found in cytoplasm. When these free ribosomes together are bound, they will be called polysome. There are some ribosomes that are attached to rough endoplasmic reticulum, and that's why rough endoplasmic reticulum synthesizes proteins with the help of ribosome. And this ribosome size comparison, we have covered this. Prokaryota has 70S, eukaryota has 80S. All right. And what about mitochondria and chloroplast ribosomes? Don't forget that mitochondria and chloroplast do have their own ribosome, and that is 70S, and that resembles prokaryotes. What are the IMAT traps? The IMAT trap, the first one is that ribosomes are not membrane-bound organelles. All right. There are some other examples that can be tested on exam. The first one we have already said, ribosome doesn't have any membrane. Then there's microtubule, microfilament, or intermediate filament. Also, you can say proteasome. And the second IMAT trap is that rough endoplasmic reticulum does not synthesize proteins directly. Okay. That means rough endoplasmic reticulum takes the help of ribosome to make proteins. And the third statement, ribosome perform protein synthesis. So don't forget these 3 take-home messages. Our next structure is endoplasmic reticulum. Let's look closely at the structure. In the structure, you see that this structure is nucleus. This is the cisternae, and inside cisternae you will find some space, and that space is called cisternal space. We represent ribosomes by these dots, and this part is rough endoplasmic reticulum. On the upper side, the endoplasmic reticulum doesn't have any ribosome. That's why this is smooth endoplasmic reticulum, and it doesn't make any protein. It usually makes hormones. Okay, now let's match the structure again. So there are 2 types of endoplasmic reticulum. One is rough endoplasmic reticulum, another one is smooth endoplasmic reticulum. The first, rough endoplasmic reticulum, it will have ribosomes attached to it. And what is the function? The function is ribosome has protein synthesis function, and that's why rough endoplasmic reticulum will also do protein synthesis, right? And it will do the initial folding of protein. Why the folding is required? Because if the proteins are not folded, then the proteins are not functional. So the initial folding will be performed by rough endoplasmic reticulum. And after that, finally, The proteins made by rough endoplasmic reticulum will be transported to Golgi apparatus via vesicles. Okay. And smooth endoplasmic reticulums, these are tubular structures and it doesn't have any ribosome at all. And what are the functions? The first function is that it doesn't have ribosome, that's why it doesn't make protein, it will make lipid and steroid. Steroid is a type of lipid, so we can easily remember that the smooth endoplasmic reticulum basically makes lipid. And also another important thing, smooth endoplasmic reticulum detoxifies the drugs and toxins, especially in liver cells. Liver cells perform so many metabolism tasks, and one of them is detoxification. That is very, very important for us to know. Okay, and then storage of calcium ions. Why calcium ion storage is important? Because without calcium ion, muscle contractions not possible. Or if we think about the blood clotting also depends on calcium. And also calcium has so many other functions in the body in keeping the cell voltage or cell potential. Calcium will also play its role. The IMAT traps, the first one is that smooth endoplasmic reticulum doesn't synthesize protein, done. Rough endoplasmic reticulum doesn't synthesize lipid. This information is also covered. And smooth endoplasmic reticulum increases in response to drug exposure. This is very, very important. There can be an example like this, that a person takes drugs and overdoses. If the person overdoses on drugs, then which component of the cell will increase? The answer would be smooth endoplasmic reticulum, because the smooth endoplasmic reticulums plays the function of detoxification of drugs. Golgi apparatus. Let's look at the structure of Golgi apparatus. Golgi apparatus. Golgi apparatus has a structure of cisternae that is kind of similar to endoplasmic reticulum. Let's say these are the flat structures. And this will be called cisternae. Okay. So cisternae has two phases. One is cis phase, another one is trans phase. So the trans phase will be far from endoplasmic reticulum, and the cis phase will be near to endoplasmic reticulum. That's why cis phase will receive vesicles from endoplasmic reticulum, and the trans phase releases the vesicles to their destination. That means Golgi apparatus does the job of packaging the proteins that were made by endoplasmic reticulums. That's why we say that modification of protein is done through glycosylation. That means glycation is being done by Golgi apparatus, and also proteins will be sorted based on their destination. That's why the particular proteins will go to definite directions, and the direction will be dictated by Golgi apparatus. And you can compare Golgi apparatus to traffic police, because also traffic police sometimes gives you the direction when needed. And packaging, we have covered that, and secretion as well. Secretion to cell membrane. And lysozyme, this organelle is basically made by Golgi apparatus. Okay. IMAT traps. Golgi apparatus doesn't synthesize proteins, and Golgi malfunction leads to secretion failure. Whenever you see that secretion failure is present in a question, you must remember Golgi malfunction. And Golgi apparatus doesn't synthesize protein, it only packages it. Lysosome. Let's look at the structure of lysosome. Lysosome has a membrane, and inside lysosomes you will have these enzymes. These are called hydrolytic enzymes. Okay. So the structure, the lysosome has single membrane. Lysosome doesn't have any double membrane, you must remember it. And it contains hydrolytic enzymes, and it is active at acidic pH. Very important to know. What is the function? The function is that all are destructive functions. That's the theme. So the first one will be intracellular digestion of macromolecules. Okay. And the second one is autophagy and recycling of organelles. Autophagy means that lysosome engulfs the damaged cell or cell organelles and it recycles them. Then lysosome will initiate apoptosis when enzymes are released. Remember, this apoptosis is controlled by mitochondria. This is a high yield information. Okay. What will be the traps or functional logics? First of all, lysosome membrane prevents enzyme leakage. That means lysosome membrane has the function of preventing the enzyme leakage. The part that controls it, this is called stabilizer. And if there is rupture, then there will be self-digestion of the cell, and this process is called autolysis. What are the IMAT traps? Lysosomes are prominent in animal cells. Plant cells don't have lysosomes that much. Plant cells use large vacuoles for similar functions. Very important to understand. Chloroplasts. Chloroplasts' function is photosynthesis. That means chloroplast will use the light energy, and with the help of light energy, it will convert the carbon dioxide into glucose or carbohydrates, and it will be stored as starch in plant cells. So the structure, if we see, then the double membrane will be present, and there will be thylakoids and grana. So the keywords are double membrane and thylakoids and grana. In this diagram, let's find out the structures. The outer membrane we see here, and this is the inner membrane. Between these two membranes, one is intermembrane space. This is actually matrix, and matrix is sometimes called stroma. So inside stroma, you will have thylakoid that look like coins, individual coins, and when the thylakoids are stacked up against each other, they will form grana together, and the light dependent phase of photosynthesis will happen inside the thylakoid. After that, the light independent phase or the glucose production phase will be in stroma. That means the food will be stored in stroma. That's why it is written that stroma contains enzymes, circular DNA, and 70S ribosomes. Okay. What are the functional logic? The light-dependent reactions occur in thylakoid membranes, and light-independent reaction occur in stroma. The light-dependent means you will need sunlight, and with the help of photon or sunlight, you will break down the water molecule, and you will have proton, oxygen molecule, and electron, and that oxygen or the energy will be used in light-independent reaction, which will be occurring inside stroma. What is the evolutionary logic? You will remember E is equal to mc squared. That's a simple mnemonic. In this endosymbiotic theory, two organelles have taken part. One is mitochondria, another one is chloroplast. That's why it is said that presence of own DNA supports endosymbiotic theory. So what is the final IMAT integration logic? Cellular efficiency depends on compartmentalization. Protein secretion requires nucleus, ribosomes, rough endoplasmic reticulum, and Golgi apparatus. Nucleus will have gene, and this gene will be responsible for making protein with the help of ribosome. And rough endoplasmic reticulum has ribosomes attached to it. That's why rough endoplasmic reticulum will make protein. Prokaryotic cells perform all life processes without organelles. That means that prokaryotic cells only have ribosome. Ribosome will make protein, and the respiration will take place on the membrane of prokaryotic cell. One of the most important topics, cell membrane structure. Before studying the description, let's look at a structure. Sometimes you need to identify different structures from this membrane. First things first, you see that it has bilayer. This is a layer and this is a layer. And this bilayer is made up of phospholipid. The head is made up of phosphate and the tails are made up of lipid. The head is hydrophilic, that means it will attract water. The tail is hydrophobic, that means that it repels water. These are hexagonal shaped carbohydrate molecules. They are on the periphery of cell membrane. And you see that throughout the cell membrane, there is a large bulky molecule, and this is a protein, and this protein is called integral protein. That means that carbohydrates are found outside the cell membrane, and inside the cell membrane you will have integral protein. Another important structure is cholesterol. Sometimes on questions they might put the cholesterol outside and they might try to confuse you. So you must remember cholesterol lives inside the cell membrane. Okay, now let's integrate them all together. IMAT will repeatedly test how structure explains functions. Cell membrane is the most tested structure function topic in IMAT biology. So the structure that we have discussed previously, it was about fluid mosaic model. Because phospholipids move laterally within the layer or bilayer, proteins are embedded irregularly. That gives its characteristic mosaic shape. The structural components are phospholipid, and the head was hydrophilic, the tail is hydrophobic. What is the arrangement? Hydrophilic heads face the aqueous environment, and the tails, it faces inward and it is away from water. What is the functional consequence because of this? Because of this, the cell membrane will form permeable barrier, and it prevents the free passage of polar and charged molecules. That's why cell membrane works perfectly as an effective barrier. Now let's discuss membrane proteins. We were studying the cell membrane, and in that cell membrane diagram we saw some proteins. Those are the membrane proteins, and that's why it is said that membrane proteins are embedded within or attached to phospholipid bilayer. If the span of the protein is throughout the membrane, then it will be integral, and if the membrane protein is surface bound, then it will be peripheral. This is very important, the cholesterol, and cholesterols are found only in animal cells, and this is located between phospholipids and it regulates membrane fluidity. What is the logic? This cholesterol prevents membrane becoming too rigid at low temperature and prevents membrane becoming too fluid at high temperature. That means that cholesterol will control the fluidity of the cell membrane. This is very high yield for our true-false type statement questions. What are the glycoproteins? If you break down the words, glyco means carbohydrate or glucose and protein means protein. So if carbohydrates are attached to proteins, that will be glycoprotein. If the carbohydrates are attached to lipids, it will be glycolipid. And these glycoproteins or glycolipids are found on outer surface of membrane, not on inner surface. Very important distinction that needs to be made. The functions are very important. These glycoproteins are like towers or radars, and it will help in cell recognition, cell-to-cell adhesion, and immune response identification. Very important for us to know. What are the IMAT traps? Cell membrane is not rigid, and proteins do not form a continuous layer. That's why it is mosaic. And carbohydrates are found only on the outer surface, not in the inner surface. And after the basics, now let's talk about the function-based logic. There are types of membrane proteins. One is transport proteins, and these transport proteins are the proteins that allow passage of specific molecules or ions and include channel proteins and carrier proteins. Okay, what are the channel proteins? These channel proteins usually transports ions, and the carrier proteins, they might carry other particles apart from ions. So what are the IMAT logic based on this statement? Transport proteins are specific. That means that one protein will carry only one specific type of ion or molecule, and each protein transports And that's why it said that each protein transports only particular molecules. Now receptor proteins, they don't perform any transportation function. They are only there for signaling purposes. Let's say this is a receptor protein and a molecule comes. When they attach together, there will be a response, and that will be intracellular response. As a result, inside the nucleus, there will be gene activation. That's why receptor proteins are responsible for gene activation. What about enzymatic proteins? The proteins that perform the function of enzymes are called enzymatic proteins because they catalyze the reactions at membrane surface. All right. What are the cell recognition proteins? They are often glycoproteins and they identify cell type, and they are also vigilant against self and non-self proteins. Because if a non-self protein invades our body, that is antigen, and it can produce diseases. That's why our body's defense mechanism is very strict when it comes to identifying or recognizing antigens. Exam logic: loss of membrane proteins affect transport and signaling, but not membrane structures. Lipid bilayer provides barrier, proteins provide function. Very important. Lipid works as a barrier and proteins provide function. Okay, now transport mechanisms. When we are studying this transport mechanism, you must remember, is there presence of energy? Is there presence of carrier protein or not? So that's why it is said that IMAT distinguishes passive versus active, protein versus not protein, and energy versus no energy. The first one is diffusion. For diffusion, you must remember, diffusion is the process where particles move from high concentration to low concentration till it reaches equilibrium, right? So this is called net movement of particles from a region of higher concentration to lower concentration until it reaches equilibrium. What are the key features? This is a passive process. That means it doesn't require any energy or protein. That's why it is said that no ATP required, and it only depends on concentration gradient. And if the concentration gradient goes away, then the diffusion will stop. That means that it will reach equilibrium. Factors affecting the diffusion will depend on the concentration gradient. The more the concentration gradient, the more the rate of diffusion. The more the surface area, the diffusion will increase. And the thickness of membrane, if the membrane is thicker, then the diffusion will go down. And temperature, if you increase the temperature, the particles will keep moving more. Let's say they are jumping back and forth, and diffusion will increase as a result. Okay, so there are some high yield examples. Let's say oxygens are entering cells. So this is diffusion process. On the other hand, carbon dioxide leaving cells, this is also diffusion process. Let's consider that inside lungs you have alveolus. Let's draw an alveolus, and alveolus surrounds some blood vessels or capillaries. The capillaries doesn't have that much oxygen, and alveolus has so much oxygen. So that means the oxygen concentration is high inside alveolus, and the oxygen concentration is low inside blood vessels. As there's concentration gradient, the oxygen will keep diffusing towards the capillary, and this is called gaseous exchange. What are the IMAT traps? Diffusion doesn't require any protein. Don't get confused. Diffusion does not require protein, and diffusion continues until equilibrium is reached. That means diffusion will not run indefinitely. Okay, what about facilitated diffusion? The main important thing to consider that facilitated diffusion will have a carrier protein. This is still passive transport because there's no expenditure of energy, but it will have transport protein. Whenever you have energy requirement, it will not be passive anymore, it will become active. The key features: no ATP required. That explains the passive transport mechanism and movement down the concentration gradient, just like before. And it requires carrier or channel protein. The molecules will sit on the carrier proteins. Let's say this is a carrier protein. Let's say this is a molecule what will happen? The protein will move, the carrier protein will move, and with the help of carrier protein, the molecule also go to its destination. Okay. What is the functional logic? There are some large or polar molecules. The polar means the charged molecules that cannot cross lipid bilayer. That's why the carrier proteins will help the large and polar molecules to bypass this lipid layer. And rate is limited by number of transport proteins. It means that the more the number of transport proteins, the better the diffusion rate would be. And what is the IMAT trap? Facilitated diffusion is passive. It is not active because it doesn't require any transporter, and saturation can occur due to limited proteins. That means that if you have little to no proteins, then saturation will happen very fast. What is osmosis? When we were talking about diffusion, we were talking about the particles, and we were talking about high and low concentration. In case of osmosis, the particles or solutes will not move. The fluid or the solvent or water will move in this case. Okay. That's why it is said that there is net movement of water molecules across a partially permeable membrane. Without a partially permeable membrane, no osmosis process can take place. And in case of this, the water potential will be high, and from that high water potential towards low water potential, the water will move. So to recap the key points, first one is that it conserves only water and solute doesn't move, and membrane must be partially permeable. These are the three most important things. Okay. And cell membrane has this partially permeable property. What is the biological significance? This osmosis maintains cell volume. How does it do it? Let's say a cell has overload of fluid, and at that moment, with the help of osmosis, the fluid will go out and vice versa. Why the fluid is only going out, not the particles? Because the cell membrane is semi-permeable, and it is essential for plant cell turgidity. Turgidity means the pressure inside cell. If the turgidity is not maintained with the help of osmosis, the cell might get broken down, or the cell might go through the process of lysis. What are the IMAT traps? Osmosis is not diffusion of solute. Remember, osmosis is diffusion of water or fluid. And this direction depends on water potential, not the solute concentration directly. This is very, very important, right? It doesn't depend on solute concentration directly. Okay. Now let's come to the active transport. Active transports will have the requirement of energy, and the definition will be movement of substances against their concentration gradient. Okay, what are the key features? Obviously, it will require ATP or adenosine triphosphate. It will use carrier proteins, and energy is also used to change protein shape. And there are some famous examples. One famous example is sodium-potassium pump. Let's say this is a cell that I have drawn, and let's say this is a sodium-potassium pump. This sodium-potassium pump will pump sodium outside, and it will take potassium inside. It will pump 3 sodiums outside the cell and 2 potassiums inside the cell. And by this, the membrane potential and nerve function is maintained. So sodium-potassium pump is very, very important. And the sodium-potassium pumps need ATP. If ATP production is lost, then sodium-potassium pump will stop functioning. As a result, the concentration inside the cell will become imbalanced and the cell will break down. This explains why oxygen deprivation prevents sodium-potassium pump function. What are the IMAT traps? Active transport stops without adenosine triphosphate. When can it happen? If there is oxygen deficiency, hypoxia, etc. And carrier proteins are specific. That means that one type of carrier protein will only perform one function. And this cannot occur without intact cell membrane. That means that the cell membrane cannot be damaged. If it is damaged, then there will be leakage and the ATP cannot be used properly in this case. Then let's come to endocytosis and exocytosis. Endo means inside cell, exos means outside of cell. That means when you take something inside the cell in bulk amount, this will be endocytosis. And when you put something outside the cell in bulk amount, this will be exocytosis. And this process will require energy, and this is used for large molecules and particles. Okay. What are the types? We have covered. Endocytosis means substances enter the cell. Exocytosis means substance will leave the cell. This endocytosis or exocytosis, this is not diffusion. This is separate from diffusion because it will require energy. And this is not protein channel based, and this is used when particles are too large to cross the membrane directly. Now, cell surface area to volume ratio. This is a classic IMAT trap. To understand the logic, let's write two formulas. The surface area is 4 pi r square and the volume is 4 by 3 pi r cubed. That means that if you increase the radius, cell surface will increase, but not as fast as volume, because when you increase the radius, the volume is increasing with cube of radius. That's why if size increases, volume increases faster than surface area. And what is the biological consequence? When surface area is reduced relative to volume, diffusion becomes inefficient because diffusion depends on large surface area, but the volume is increasing more compared to surface area. That's why the surface area will not be sufficient to cover the large volume. And that's why the cell becomes small. Because if the cell becomes large, the volume will increase and waste removal and nutrient uptake will slow down. And that's included in this biological adaptation. And this explains biological adaptation. Cell divides to reduce size and folding of membranes are there, such as cristae, which is found in mitochondria, and microvilli, this is found in intestine. And cell shapes are thin or elongated, because if the cell shapes are greater, because if the cell shapes are big, then because if the cell shape becomes bigger, then the volume will increase and that will hinder the normal process. So IMAT logic, cell remains small to maintain efficient exchange. That means diffusion. Large organisms are multicellular and not large cell. So what is the final IMAT integration logic? That membrane structure determines permeability and transport mechanisms depends on energy and protein. Cell size is limited by diffusion efficiency, because if the cell size becomes very large, then diffusion becomes very difficult. If you revise only what we annotated and drew today, you are fully prepared for IMAT cell biology. But before going, we will practice some questions. The first question is that which structure is present in both prokaryotic and eukaryotic cells? Obviously, it will be ribosome because both the cells need protein production. Sometimes some people get confused and say that DNA, but DNA is not an organelle. DNA is just chemical structure or molecule. The second question, which statement about mitochondria is correct? Mitochondria is actually only found in eukaryotes, and mitochondria is responsible for respiration. So the first one, found in prokaryotes, this is false. Site of photosynthesis, no. Actually, chloroplast is the site of photosynthesis. The third option, they contain their own DNA. This is true, and their DNA is circular. And option D, mitochondria is not single membrane organelle. It has double membrane, and it doesn't produce ribosome. This is false as well. Ribosome has rRNA and protein inside it. Okay. And what about question 3? Rough endoplasmic reticulum is mainly involved in what? Rough endoplasmic reticulum has ribosomes, and that's why it will make protein. The answer would be B. What about the next question? Which component makes the cell membrane selectively permeable? Obviously phospholipid bilayer, because the phosphate head is hydrophilic and the lipid tail is hydrophobic, and this allows selective permeability of the cell membrane, and it also protects the cell from harmful components. Question 5. Which process requires adenosine triphosphate? Obviously, the active transport. Diffusion doesn't need it. Osmosis doesn't need it neither. Facilitated diffusion needs transporter protein or carrier protein, not the energy. Simple diffusion, it doesn't need any ATP or energy as well. Question 6. Osmosis is best defined. Remember, diffusion means movement of solutes, but osmosis means the movement of water molecules. That's why the answer would be water molecules. Question 7. Which transport process uses a carrier protein but no energy? The answer would be facilitated diffusion. Remember, diffusion never uses energy, but as there's facilitated word attached to it, that means that it will use transport process or transport protein. Question 8. Increasing the surface area to volume ratio improves what? DNA replication? No. DNA has nothing to do with surface area. Protein synthesis? No. Protein synthesis is done by endoplasmic reticulum or rough endoplasmic reticulum. Diffusion efficiency? This is important and this is correct, right? Because the increased volume is not good for the cell. The surface area, if you increased, then it helps in diffusion process. Which structure modifies and packages protein? Remember, ribosome makes protein. Lysosome is involved in phagocytosis or eating up of cell or debris. Golgi apparatus does the packaging of protein. It also gives direction for the protein to go to its destination. What about nucleus? Nucleus doesn't package protein. Nucleus gives the genetic code. What about smooth endoplasmic reticulum? Smooth endoplasmic reticulum makes lipids or steroid. Also, it can help in detoxification or calcium ion storage. Then which statement about lysosome is correct? Lysosome is found in plant cells. This is incorrect. Lysosomes are mainly found in animal cells. Option 2, involved in protein synthesis. This is not true. Option C, contain digestive enzymes. This is correct. Site of respiration? No, lysosome doesn't perform respiration. Respiration occurs in mitochondria and in case of prokaryota, cell membrane. And lysosome is involved in photosynthesis. This is not correct as well. And these are the solutions given. And these are the homeworks, because if you can perform the homeworks correctly from past papers, then it will become very easy for you. The first practice is that all practice questions of prokaryotes and eukaryotic elimination questions, then membrane transport comparison tables, and also try to draw and label phospholipid bilayer or phospholipid bilayer of cell membrane and mitochondria. And after that, answer this question in one sentence: why are mitochondria absent in red blood cell? I will give you a hint, because red blood cell needs to accommodate hemoglobin and carry oxygen. In the next lecture, we will cover enzymes and metabolism. Thank you very much for watching. Hope you liked it. And if you liked my lecture series, then you can buy my ebook about complete IMAT preparation, and the links will be provided below.